ado, I'd like to introduce Danny Chou from University of Utah to talk to us about monomeric insulin.
Hi, I would like to thank the organizers for the invitation, and it's very good to be here again. I'm from the other side of the mountain, but both sides are very different. So my lab started about 2 years ago, and we are more academic focused, but I hope to kind of introduce some of our work to you, and then today more of the monomeric insulin part, and hopefully it won't be too boring for you guys. So my background is more about small molecule total synthesis and MACAM screening. That's my PhD work, and then I got introduced into this peptide world, starting my postdoc, and it has been pretty good, and then I kind of stay in this world to continue my independent lab. So this is all about insulin, and then I guess because of insulin is such a fascinating molecule, and then everyone in the peptide field will know about it. So basically, as you can see, insulin is a hexameric form. Each color here represents one monomer, and then you can see that basically two insulins will bind together to form a dimer, and especially you will see this anti-pyruvate interaction to be the dominant form. And then in the B10 histidine, 6 insulins together using 2 zinc ions, they will form this very stable hexameric form, and then it's the same form that got stored in beta cells. So the problem of insulin multimerization is that when you do subcutaneous injection, you tend to have those delayed time in terms of the body absorption. So basically, you have to wait until insulin becomes monomer to have the strongest penetration to the body. And as I said, this is pretty much well, there are multiple dimer formation regions, but it's pretty much dominated by this Phe aromatic triplet, and then you will see in 2 different insulins, and then form an anti-pyruvate interaction, and then in a way that kind of to dominate that. So this is kind of an old review now, but still, I mean, I would say insulin dispro and insulin aspart. They are the 2 dominated fast-acting insulin in the market so far. And as you can see, basically, they are just simple amino acid modifications in this C-terminal of the insulin B chain, and then they tend to reserve the activity of the insulin. And then we will talk a little bit more about, it's not too much. Okay. So in the case of insulin aspart, what it does is there's a simple mutation from protein to asparic acid in the B28 position. And as you can see, this interaction will create these repulsive amino acid pairs in the dimer formation region. So although those aromatic triplets, they are still here and then to have the dimer kind of interaction, but due to these 2 pairs of repulsive pair, you can have more and more kind of monomeric form than just the pure dimer form. So you can see that it's an old paper more than 20 years ago now. So they can see that you can design different things and then try to control the association state. So in a human insulin, you will be a hexamer. And then this is a pretty famous X10 molecule. You change the histidine, which is required for the zinc chelation, mutated to asparic acid, so you can block the hexameric form, and you can see they are more or less dimeric. And then you can see that when you make it lower and lower, and in this case, this is the aspart, so you can see they go to more and more monomeric state, and from the initial absorption rates, you can see that they are reversely correlated with the association state. So this kind of tells us that well, in a kind of high-level way, you make it more monomeric, hopefully you can get things absorbed faster. So this is also a very exciting time because of a potential development of artificial pancreas. So basically, I would really say this has nothing to do with the drug itself, because basically CGM or continuous glucose monitors, they were approved some time ago, and then insulin pump has been approved for some time, and then these engineers, they just thought that, you can just use an app to do the algorithm to control the delivery of insulin or glucagon as a way to mimic pancreas. So as we know in the normal body, what it does is alpha or beta cells, they will sense the glucose levels, and then you go do a whole signaling pathway to secrete insulin or glucagon as a way to maintain the blood sugar level. But in this case, what you get is instead of alpha and beta cells to sense the glucose levels, you have continuous glucose monitors to give you the trend, and then based on the trend, and then the algorithm here to kind of tell pumps to inject either insulin or glucagon to do the job. So it's pretty easy for them to do their trials because CGMs were approved, pumps were approved, and then there's no toxicity from the app, so they can start to do phase 2 directly instead of doing phase 1. So basically, I would say there are multiple groups doing these kind of studies, and then words are, seems like they will get approved next year or the year after. I first got kind of introduced to this concept, and then that was from this guy who's a professor in Boston University. His name is Ad Emanuel. He was working on something totally different, more like wireless thing, but his son has type 1 diabetes, so he started to kind of shift his own focus to work on that. So I still remember at that time, that was still when I was a postdoc in MIT working for Bob Langer. He gave a seminar at MIT, and then he was talking about this concept. I remember he mentioned this thing, and this is actually the same sentence is in this New England Journal of Medicine paper. He is complaining about, well, as an engineer, not as a chemist or biologist, he is an engineer complaining about that the current available rapid-acting insulin is still not fast enough for them. So they got lots of kind of difficulty in terms of designing the app, the algorithm to do a better job. So, you know, although our pancreas can do the job pretty well, in this kind of arterial pancreas, it's still pretty hard. I think in their case, they are using Dispro, although that's, you know, Dispro and the aspart is the best available right now. They still think it's not good enough. So that's kind of made me to start to think about, you know, can there be any kind of truly monomeric insulin that can be used, and basically you can, you know, still have the activity, and then potentially that can help to be a potentially ultra-fast insulin. So I kind of start my lab around this concept. And then one thing you can think about is to treat it with trypsin, so you can cut this whole A amino acid off, and then this is a pretty famous human shortened insulin called DOI, disoctapeptide insulin. So this insulin can be potentially monomeric because you pretty much cut the main dimer formation region off, and then, you know, you do some ultrasonic centrifusion, and you can prove that it's indeed truly monomeric. So it is, but the problem is this guy is not active at all, if you want to consider. So basically, I think in the old days, so this is still from 1980, more than 30 years ago, but in the old days, like those papers in 60s and 70s, sometimes they see, you know, 10%, 15% of activity related to human insulin. Sometimes they see 100-fold lower, but I think this is the first paper that really definitively showing that it is at least 1,000-fold weaker in terms of human insulin receptor binding and the activity. This is because I think in the old days, the column chromatography technologies are not as good, and then they have a hard time purifying DOI from human insulin. So the residual one give them enough activity. But I think this paper, they use lots of different methods and then trying to purify it. So they prove that, you know, there's at least 3 orders of magnitude off in terms of the activity. So basically, this is not active, and this is not, well, now we know that it kind of makes sense. So Mike Lawrence and the Michael Weiss, they kind of have the structure of insulin and some micro receptor, so some subunits of the insulin receptor. So they kind of prove that right now there is a conformational change in the C terminal of the insulin B chain, and basically this phenylalanine 24, B24 in human insulin is pretty important to fit into the pocket. So basically, in the case of DOI, there's no such thing because we cut a whole segment off, and then we do not have that group to bind with the receptor, and hence you see a 1,000-fold decrease in affinity binding. So, this kind of like well, to me at that time, it's kind of like a dilemma. Like, you can cut the whole thing off. It can be truly monomeric, just like monomeric, like no dimer formation at all because there's no dimer formation region, but you just cannot get the activity. So we start to think about what we can really do. So I would like to show you a picture about a movie about corn snails. So these are snail animals live on the bottom of the sea, and they eat fish for living. And then you will feel that, it doesn't make sense because snails swim so slow and fish, like this guy here, they swim so fast. And there's no way snail can eat fish. But as you can see, snail just opens its mouth and fish just stay there. And the snail just eat it. Okay, just eat it. You can see slowly, gradually, but eat it. Okay, and you will feel this very fascinating, but you will probably still think why a chemistry faculty in academia want to talk to you about how corn snail eat fish. But that is, as you can see, it's pretty bad. That is indeed what we want to talk about. Oh, I cannot get it. It doesn't want me to stop it. Can we go to the next slide? Okay, so we want to talk about how corn snail eat fish. Okay, so you will see basically snail here, you got lots of fish there, and then snail just opens its mouth, fish just still stay in the same motion, and corn snail just eat it. Okay, so there are people fascinated by this process, and they went back to study, what's inside it. So as you probably know about corn snail, there are about chronotoxins. These are relatively short peptides, I would say generally 30 to 40 amino acids. They have pretty linked disulfide bonds, okay? But in this case, they found a molecule like this, okay? And then actually, this guy, his name is Toto Oliveira, so he's in the biology department in our institution as well. When he came to me about this, and I say, this looks like human insulin. Just do the disulfide bonds linkage here. But you can see that there's still something different. The different parts here, the dimer formation part is gone. Okay, so this is a way shorter insulin, and actually there are only 43 amino acids there, so it's lacking 8. Basically, you got one fewer amino acids in the H end in the C terminus, and then you pretty much have this whole C terminal of the B chain segment gone. So at that time, I told him that I will bet my money with you that this guy will be monomeric. So this insulin is quite interesting. So they published it last year in PNAS. So this insulin is short, but I think the most interesting here is it has 4 post-translational modification. And I would say that in the evolution, in the case of insulin throughout the evolution, we don't really see PTMs here, but PTMs are pretty common for chronotoxins, especially for those kind of peptides that were secreted through the venoms. Okay, so there are 2 gamma-carboxyglutamate acids. There are one in A chain and one in B chain, and then this one is actually in the related B10 positions in the human insulin. So basically, in human insulin, this is a histidine for the hexamer for formation to chelate with zinc, but it's not here. There's also 1,2-hydroxyl protein here in the B chain, and there's another very funny thing is this is actually an amide bond in the end of the C terminus of the A chain inside of the normal carboxylate. Okay, so this insulin is not isolatable. So basically, the way they figure this out is do metagenomic sequencing, and then they kind of guess what's there, and then they use mass spec to detect the line. So this is not abundant, so they have to do some chemical tyrosines to do it. When they first start the study, they have a hard time trying to make it. So what they make is a diselenium form. So basically, the way they do this is because if you have a diselenium bond in this intramolecular to replace intramolecular disulfide bond, it's easier for their synthesis. That's because you can avoid potential disulfide bond shuffling here. So they first make the A chain, but in this intra one, they have selenocysteine to make it, and then they make the B chain, and then basically with two free cysteine, they combine it, and then also you use the simple kind of the well-known iodine, and then to form the third sulfide bond. So in this case, they call it S-con-ins1. So the S means the selenium bond. Okay, so they do the study and then try to see whether it's useful or not. And for them, they try to sell the story, and then this is the insulin that fish was killed by snails. What they do is they have zebrafish here, then inject STZ into the zebrafish and to induce diabetes. And you can see that for these diabetic fish, they have higher glucose levels. And then when you inject either human insulin, they have some activity, or this selenium form of the snail venomous insulin. You can see the blood sugar level go down. And here you can see that this is the baseline. Fish is moving. This is with the insulin, probably due to the hypoglycemic shock. So you can see that they don't move as fast. So in this case, well, to our collaborator, Toto Oliveira, this is kind of a good story for them. They discover a potential insulin from the snail venom, and it can have effect on fish. So story ended. So, they are happy. But the thing is, there's really not a lot of value to try to cure diabetes in fish. So we try to answer whether, there can be any human implication or not. So you probably need to help me to move to the next slide. There's some problem about the video ones. Next slide. Okay, so for us, when we first started the project with them, we tried to see whether we can make a real form of this mammalian insulin called INSG1. So basically what we're using is very similar to what people do with the human insulin, but we actually got lucky on this. That's because you can see there are lots of charged residues in this insulin A chain, as opposed to the human one. So in human one, lots of times you need to have special strategies, but in this snail one, it's actually pretty straightforward. So basically, we can just use the ring amide to make the A chain, so that will give us the amide bond in the end. And then basically, also it's pretty simple. Pretty straightforward strategy that same thing. You form the first bond, the first disulfide bond first, and then you leave it with this, and then you cleave it with the A chain, and then you make the B chain, so the B chain will be the same way, but you activate the other cysteine here, and then basically you form the first bond. That's pretty easy, and then the iodine one form the second bond. So the overall yield is somewhere around 5 to 10%-ish, carrying from the A chain, but for us, this is good enough. So we start to study this, and the first thing we want to study is to prove that whether it's really a monomeric form, and it is. So basically, we do the AUC, and then we got a molecular weight, and then if you calculate based on the snail insulin sequence, it will be somewhere around 1.05 association state. So it's pretty monomeric. So the next thing is we want to ask whether this can be potential, mammalian activity at all. So this is a sequence of the constant insulin. This is a sequence of the human insulin. So you can see that the constant one is way shorter, and then the constant one looks more like the fish one. Okay, so basically this snail insulin, although we call it snail insulin, but this is in the venom, okay? And then snail has their own endogenous insulin. And this venom insulin is closer to the fish one. It has nothing to do with the endogenous one. So you can see that basically, because there's no such fragment here that is required, especially this B24 phenylalanine, you will guess probably it's not going to bind with the human insulin receptor. So I'll point out two key things here. This is the key thing that we just talked about, and then also normally, through the literature, you would think that the A21 asparagine is pretty important for the binding, especially for the insulin receptor binding site 1. And this is suggested by Pierre Demetz, close to 40 years ago now. So basically, you can see that these two insulins, if you cut the A21 off, or you cut the whole the C terminal of the B chain off to get the DOI, you can see that they are way off the chart. So basically, you know, they don't really bind with the receptor, and then they don't really induce the negative cooperativity in insulin receptor. But we do it anyway, and what we see is actually quite interesting. So in this case, this one is a human insulin receptor binding. So this is a human insulin. The blue one is the real venomous insulin. So you can see that it's kind of, in this case, 34 weaker in the, you know, the activation part is 24 weaker. So you can see that it's actually kind of modestly active. And then I have DOI here as a comparison. You can see that it's way better than DOI, although still, I mean, it's not, you know, as active as you would think. But still, this is very interesting in the way that, you know, you will guess that it will be very similar to DOI, but in the end, it is not. It is somewhat active. And then we further try to see whether the PTMs are important. So we make one analog in a way that basically you have these to be acid, and then you have these two to be glutamic acid, and this one to be protein. And what you see is you lose the activity by another 15-fold-ish in the activation part. So basically, this is telling us that the post-translational modifications are somewhat important. And this is the only, you know, easy animal study we have. So we do a simple ITT here. We cannot inject the normal dose. We inject for the human insulin because it's not potent enough, but we inject 2x, when we inject 10x of the dose, you can see the glucose level start to go down in STZ-treated mice. So the real question here is, you know, it's actually not that bad, but why, right? Because you will imagine without that fragment, without A21, it should be pretty bad. So we start to work together with Mike Lawrence, try to solve the structure of it. So the paper just got published 2-3 weeks ago in Nature Structural Microbiology. So basically, we try to know what's going on in this snail insulin, and then what can we really learn from it. So the first lesson we learned is its backbone between the snail insulin and the human insulin. They are very similar. They have nearly identical. I mean, I would really say that probably just because the B chain of the human one is longer. And they have a little bit different size in the A chain, but other than that, they are pretty much the same. But this is actually not too surprising, I would say. That's because all different kind of insulin-related structures, they all have somewhat similar backbones. You know, it can be insulin or IGF-1 or even the fly insulin. So this structure was, I believe, it was solved by Novo 5 years ago. And also some other, you know, insulin-related proteins that are not really insulin. They all have the similar structure. So you would not guess this is, you know, too surprising. The second lesson we learned is we still don't know whether PTM can really do anything with the receptor itself, but we do see some more interaction due to PTMs to interact with the backbone and then to create more stability for the snail insulin. And this is quite interesting. The third lesson we learned, this is actually, I will say this is probably the most important one, because why there's no B24 phenylalanine in this cone snail insulin, but we still see some activity. And what we end up seeing is there are two mutations here in this venomous insulin. B15, which is normally a leucine in human insulin, B20, which normally is a glycine in human insulin, they got mutated to tyrosine. And then we believe that these two tyrosine, due to the proximity, may contribute to the binding pocket of the B24 phenylalanine in the case of human insulin. So, but they are not perfect in the structure. In this B15 tyrosine, it normally goes this way. So we suggest that maybe there will be a rotation of this residue to fit into the group upon receptor binding to fit the pocket. And in this B20 part, we actually don't know too much. That's because in this part, the resolution of the crystal is pretty bad. So basically, it can pretty much go anywhere. And then as you may remember, there's nothing here. So basically, this is pretty free. So it can move to anywhere. So we think that there's a probability that this B20 tyrosine may move in to fit in the pocket. Okay, so this is what structure suggests, and this is the end of the paper. We didn't really try to, in the paper, we didn't really try to answer to see whether it is true. And then this is a very important question. So we try to answer. The first thing we do is we actually make mutants on these two positions. So we just do alanine scanning to put them into alanines. And what we see is, yes, it does do the activity. So basically, you can see that they are closer to DOI now when you take these two away. So this kind of proof that, these two tyrosines are important, and then it can be potentially useful to compensate the role of the B24 phenylalanine. But still, at least to me, the goal is whether you can use this same strategy on DOI, which has no such fragment and no B24 phenylalanine, and make DOI to be somewhere, closer to insulin to activate the insulin receptor. And as I said, so basically it was originally leucine here and the glycine here. And as you can see, these two positions, they are not, at least in the case of just a receptor residue mapping, they are not known to bind with either receptor binding pocket 1 or binding pocket 2. Okay, so this is kind of interesting for us to see whether we can see any effect. So we decide to start to make it, and we use the famous Value paper at the time when he was still in Lilly, and then basically we use the same strategy. This is because the insulin A chain is pretty bad, and basically the take-home message here is they introduce iso-ACO peptide TS here, so basically you can introduce one more amine group in the chain to increase the solubility. So we pretty much use the same strategy. The only difference is we have a shorter B chain here, and then we can just start to mutate different positions and introduce stuff we want. So we make human DOI in a way that we put one tyrosine on the B15, on the B20, or we put it on both. And then you can see that I would say the effect is relatively subtle and modest, but you can see a significant increase in terms of that. The B20 tyrosine is more important than the B15 tyrosine here. So basically, you can see it in a way that you can increase the effect somewhere between 5 to 10-fold. It's not perfect, but at least we are seeing something in it. To us, at that time, it's pretty exciting. So, but as for our goal, whether human DOI can be as active as, not just talk about human insulin, just talk about the snail insulin. Here is still pretty hard to know, but we believe that because snail insulin does not have that fragment, and in the shorter form of the human insulin, we can probably still do the job. So here, I'm going to show the last data slide. So I am not allowed to show you the sequence here, but basically what we can see is we can indeed make DOI to be as good as snail insulin, and actually better than the snail insulin now. So basically, these are like purely MECAM protein efforts to change the sequence of DOI. So you can see that we have the series analogs here. The green one is the snail one. So some are better than the snail one, some are weaker. So now we are somewhere around 10-fold away to do it. And then basically we just try our best and then to further improve the potency. So where are we going right now? So we have this wonderful collaborator, Toto Oliveira. They study way more corn snails, and there are just one species. There are way more, and then they just published a paper in microbiology in the evolution. There are way more weird insulin-like sequences from corn snails, and we are trying to see whether we can make it. And then as you can see that some of them are really short, some of them are really long, and then some of them got a longer A chain, some of them got really long B chain as well, but in a way that look very, very different from human insulin. So this is one direction for us to follow as well. Okay, so summary. So we and our collaborators discovered a venomous insulin from this species Conus geographus. It is still the smallest insulin discovered to date, and it has pretty modest potency toward human insulin receptor, and it can give so we can show in vitro, in vivo activity. But I think the cool thing is when we solve the structure, and it suggests that either B15 or B20 tyrosine can compensate the role for the B24 phenylalanine, and we show that we can indeed imply this position to DOI and then to further improve the receptor binding activation. And then the last point is kind of purely, through our academic form. So, this is pretty fascinating because insulin was discovered more than 90 years ago, and still right now, we are still seeing new sequences, and then we can still learn from and then try to, potentially, well, for us, it's still more in the pure academic point of view, but there are still lots of things for us to learn and then to do stuff. Final slide. So thank you for these organizations to support my lab, and then actually these organizations, they do not support any of this work. It was this nonprofit foundation to support it because people don't like this idea yet. And these are people in our lab. They are doing the work. Majority of work are done by Maria, who is a graduate in my lab, and Xiaochun, who is a postdoc in my lab. And then our collaborators, Helena and the Toto, they are the ones that work on the corn snails and then to discover the sequences. Mike Lawrence, the world-renowned structural biologist, he's the one to solve the structure, and then he's solving more structure for us now. And then Ray Norton more for the kind of biophysical studies. And thank you very much, and I'm happy to take any questions.